Okay, so just to summarize the photoreceptors, again, we've got cones and rods are kind of classic photoreceptive cells. They're located at the back or most, posterior or most caudal aspect of the retina, layer of the retina. And they detect photons of light. We're describing light in the form of particles, but they also, absorb, electromagnetic radiation within, specific wavelength ranges. And they include both the rod cells, which have far more extensive surface area for absorption. They have many, many more of these membranous discs that are located in the outer segment here. It's much more extensive, much more surface area, much more pigment. The pigment is a pigment called rhodopsin, which will absorb light, of any wavelength within the visible light spectra. So basically, the rod cells have very, large surface area for absorption. So they're very, very good at responding in low light circumstances, right? So they're good for night vision, for vision, at dusk. It turns out that these rod cells, too, are, many, many of them are going to be linked to bipolar cells. And the bipolar cells that they're linked to tend to be large bipolar cells. They're actually called diffuse bipolar. They have lots of projections out here. So you can imagine we've got, rod cell, rod cell, rod cell, rod cell, rod cell, all connected to one big diffuse bipolar cell. And that'll connect to one great big ganglion cell. It's called a cellular ganglion cell. It's a great big M ganglion cell. Its axons will leave the optic nerve, but it's gathering information, right, from many rod cells that are splayed over like a larger region of the retina. So it turns out that if something moves across multiple rod cells, it's more likely to be captured, by the pigment, by these discs, and have them, stop firing and remove inhibition, right? Stop the paradoxical dark current, so it's more likely to set it off if something's moving across. So it turns out these rod cells, many, many of them, linked up together to one big diffuse bipolar cell, which is linked up to one great big M ganglion cell, magnocellular ganglion cell, tend to be found in the periphery of the retina. They're off to the side here. If I'm looking straight ahead, most of my rod cells are off, not where I'm focusing my, vision on. And they're very good in low light conditions, right, at dusk, for example, and they're very good at detecting motion, things moving off to the side. So think a little bit about that. Why is that a good architecture, a kind of functional architecture for, our eyeballs? Our eyeballs are not all the same, because in the middle of the retina, right, where I'm looking is kind of a pitted structure. Like, remember you've got the photoreceptors, right, they're in like this, connected to the bipolar cells, which are connected to the ganglion cells. Well, at this kind of pitted region where I'm focusing right now to be able to see details, like on the screen, for example, this is called the fovea, and these photoreceptors are there. They tend to be cone cells, densely packed in. So many different cones, lots and lots of cone cells densely packed into the fovea, and little bipolar cells that are linked to smaller ganglion cells that are called parvocellular ganglion cells, or P ganglion cells. And also, all of that architecture is sort of splayed to the side so that the light that's coming in, that it's been bent by the lens, right, it's working its way towards the retina, doesn't have to pass through ganglion cells and amacrine cells and bipolar cells and horizontal cells, and before it actually arrives at the membranous discs and the pigments of the photoreceptors that are located towards the back. So the fovea is where you're going to find cone cells, and typically and densely packed, and not many cone cells connected to one bipolar cell, but much more one-to-one output basically from the eye at the level of the fovea. So we tend to have very good high acuity vision, very good detailed vision, lots of pixelation, basically at the fovea. Plus we have these cone cells, and there's more than one kind. In most of us, we have three distinct types of cone cells. They differ by which, pigment gene they actually express, because there's three different, pigments typically that are expressed in three different kinds of cones. And the L type, will actually absorb the long wavelength, which is typically correlates to sort of a perception of red light. It's the red end of the visible light spectrum. There's the medium, length, the M type cone cell that typically correlates with green, in terms of our perceptual experience. And then there is the short wavelength, or the S type, and that typically correlates with a perception ultimately of blue. And these are tightly packed in, and this is these are labeled lines, as we talked about, right? However, we can see not just green, red, and blue, but we see an enormous spectra of color, right? And that's because it's the pattern of activation across, these various cone cells, that matter, in terms of directing, input from the eyes ultimately into networks in the brain that allow us to perceive, this vast, number of potential colors. The other thing that's kind of interesting, I think, about these cone cells is that the red and green, we tend to have a lot more red and green cone cells than we do the blue. The blue are less numerous. And if you look at holiday lights, for example, if you're looking at the blue holiday lights, they always look a little bit blurred to some extent versus the crisper, kind of red and green. In addition, our eyes, there are glial cells in the retina. This is really fascinating. There's this set of glial cells called Mueller glial cells, and they actually act as fiber optic cables. So they tend to channel specifically the red and the green wavelengths, the medium and the long wavelength, radiation, down, through the various layers, basically of the retina, directly to, L type, the red responsive pigment cone cells, and the M type, the green responsive pigment subtype. So we exaggerate red and green, and we scatter and don't detect nearly as much of all the blue, the short wavelength, visible light that's actually out there and, trying to get in, to our retinas as well. There's also something really fascinating about cones is that, you could lack a cone, right? You could be a dichromat as opposed to a trichromat. You could, perceive, detect actually, fewer discriminative, kinds of spectra within the range of visible light. So you don't have as many, cells that can be responsive to generate complex patterns. So you may see fewer colors. And there's a test you can take here on our site called the Ishihara color test, where you could see if you could see numbers and letters that are placed within these plates of various colors. But in some of us, actually, it turns out that some of the genes for pigments, the gene for the L type, the red pigment, is actually on the X chromosome. So we can inherit different versions of these genes for the various pigments. So we can inherit maybe two different types of gene for the L type pigment. If we have two X chromosomes, so if you have two X chromosomes, you may actually have a fourth cone photoreceptor, one that absorbs maximally in the L range, red range, basically, but another one that maybe absorbs in a slightly different range maximally. So that gives you a fourth cone. So, there's some of us out here that have perhaps, a fourth cone and a more complex pattern and can potentially see, finer gradations, between in terms of our perception of color, basically in the world.